Morning, everyone. Let's get started. We are going to be doing lecture 3 today, which is about cells. So we actually have to do a little bit of note taking today. So I hope you have some papers with you. Some colors would be nice as well, okay, because we will be drawing quite a bit. All right, let's start with a little bit of an introductory background information. Previously, we were talking about the hierarchy of organization, and we all know, we should know by now, that everything that we know that exists in the universe are made up of elements, and the elements are made up of atoms, right? They are the building blocks of everything that we know. And in lecture 2, we learned how atoms can form molecules, and we spent quite a bit of time talking about the 4 major macromolecules in our body. Okay, of course, the quiz is on Monday, and on the quiz, you are responsible for the carbohydrate as well as the lipids. Okay, and of course, everything in lecture 1 as well. So now we are moving up this hierarchy of organization. We will be focusing on organelles as well as cells for today. In fact, there are a lot of things we need to learn about the cell that will carry us all the way to lecture 9 in the course. And then in the final lecture, lecture 10, we will be talking about tissue, which will serve as a nice transition into your next semester stuff. So how big exactly is a cell? Well, a cell on this scale is usually ranging, it's measured in micrometer, by the way. That's what the is. And in case you forgot, 1 meter is equivalent to a million, 10 to the 6 micrometer. And so we tend to measure cells within 10 to 100 micrometer range, depending on what kind of cell you're looking at. Some cells are bigger than others. Even within our own body, certain cells are longer, or they might be a little bit smaller, depending on what organ you find them in. Okay, plant cells sometimes are significantly bigger than animal cells, and so on and so forth. Now, inside the cell, as we will see, there are many organelles. Okay? Organelles are tiny structures that are inside the cell, and they perform specific functions for the cell. Okay? Those tend to be quite small. Okay? We're talking about 10 times smaller than the lower end of what a cell would be. And they are comparable to the size of a bacteria. We go further down in the size scale here. Viruses are measured in nanometers. Nanometers. Does anybody know how many nanometers are there in a meter? How many zeros? What's that, sorry? 4. Okay. Anyone else? 9 zeros. Yeah. Okay. It's a billion nanometer. Okay. So that is very, very small. And of course, these things are so small that it's not possible for us to see with our naked eyes. And cells, they do look quite different depending on what organism we are looking at. Over here, we have some bacteria, and not all bacteria look like this picture. These ones, they form little balls, and it's an example of Streptococcus, and which can give you like strep throat, right, for example. Over here, we have another thing. This is called an amoeba. An amoeba. Where do you find amoeba? Have you guys heard of this before? Yeah, Josh? Sorry? Dirty pools. Yeah, so sometimes they could find themselves in swimming pools, and kids, if they get it into their system, they can get diarrhea and sick. But naturally, these are found in fresh water. Okay. It's actually not a virus. Good question though. Not everything is plant or animal, there are other kingdoms as well. So this belongs to something called protist. Okay, protist. Yeah. So a lot of protists in fresh water. And then how about these ones? You guys must have seen something like that before, right? In movies and whatnot. What it's in our body. Which part of the body would you think this is? The brain, that's right. Okay, and these are the neurons, right? These are neurons. So they all look quite different, but nevertheless, they are all cells. And how do you define a cell, right? It is the smallest unit that still retains all the properties of life. Right? We learn about the properties of life in the first lecture, right? The ability to maintain homeostasis, there's energy requirement, so on and so forth, right? So all those characteristics are still retained, they are still true on the cellular level. If you go any further into the cell, if we take an organelle, for example, then the organelle is no longer alive because it doesn't have all those properties, okay? So this is as small as you can go, and you can still define it as a living thing. Okay, so of course, we are all made up of cells. How many cells do you think we have, guys? A lot, okay? Any number range? What are we talking about? Millions, billions? Okay, so have a number in your head, and we'll talk about it in a second. So even though all the cells look quite different, they all have some common features. Okay? Regardless if we're looking at a bacterial cell, human cells, plant cells, fungus cells, it doesn't really matter what kind of cell you're looking at. They all have these three features in them. The first feature is a cell membrane. It's not the first time we talk about cell membrane. You can think of the cell membrane as the edge of a cell. It surrounds the cell, it protects the cell. And another name for the cell membrane is the plasma membrane. Does anybody remember the major component of a cell membrane? It's on your quiz, you should know. Good try. That's a cell wall that you're thinking of, yeah. Not quite. Major component of the cell membrane. It's a type of a lipid, guys. Oh. Let's see, you have it? No. Oh dear, you guys have you have to study, okay? Phospholipids. Phospholipids, okay? Phospholipid is the major component of cell membrane. If you haven't already, please watch the review video, okay? You need to watch it. Okay, what else are common for cells in general besides the cell membrane? Well, the inside of the cell membrane is kind of like a gel-like fluid, and we call that the cytoplasm. Now, if you're having a hard time visualizing this, just imagine getting a Ziploc bag, fill it up with water, close the bag, okay? The water inside is like the cytoplasm, okay? And then the Ziploc bag itself is like the cell membrane. And once again, cell membrane is made up of phospholipids, primarily. And the last common features of all cells is that they are going to have some sort of genetic material. Okay, so I'm just going to draw them here as little bundle of threads here. That's the genetic material. What is our genetic material made up of? Which macromolecule is it? DNA, right? Okay, that's right. It's an example of nucleic acid, right? Nucleic acid. Hello? All right, now that we are all kind of here, I think majority of the people are here already. If you go to the, like not now, but like if you go to the course homepage later, I posted the concept track question answer for lecture 1 and 2. Okay, remember we didn't have time to do some of them, right? So download it and it will look something like this, okay? So it includes all the answers. You're welcome. All right, now we're going to talk about the cell theory. Cell theory. There are 4 parts to the cell theory. The first part of the cell theory is that every living organism consists of 1 or more cells. We kind of know this already, right? From our first class. All living things are made up of cells. But how many? Well, at least 1. And it could be a lot more. So here is another cell that's related to the amoeba. They are both protists. You don't have to know the name. And this guy is called the paramecium. Now, you will see the paramecium come back a few times throughout the course. In fact, when we do the microscope lab, right, you might be able to see the paramecium under the microscope. Okay, so they are made up of just 1 cell. They live in fresh water, similar to what the amoeba does. This is called a C. elegans. You don't have to memorize the name. C. elegans is kind of like a worm-like organism, and it's a model organism. Okay, there are several model organisms that scientists use to do research on. Right? We know a lot about these model organisms. C. elegans is an example of that. Fruit fly is another one. Mice is another model organism. So before you can test a new drugs on people, you usually test it on some of these model organisms first to see what kind of interaction biologies there are before you can move up. So this thing has 3,000 cells. Okay, and for humans we have trillions of cells. Trillions. Okay. Of course, as we grow, we get more and more cells. Okay? We have a lot more cells now than when we were first born. Okay, but once you reach adulthood, it's pretty much constant. Okay, because you do make new cells, but old ones die as well, right? Okay, so it's not like we keep on getting more and more and more. So that is the first part of the cell theory. Everything that's alive, it's made up of 1 or more cells. Part 2 of the cell theory states that a cell is the smallest unit of life. And we said that already. It retains all the properties of life. But more importantly, they are individually alive, even though they are part of a multicellular organism. Let me just go back here for a second. If something only has 1 cell, then we actually call it unicellular. If it has more than 1 cell, then we say multicellular. Okay. Sometimes it's hard to believe we are made of trillions of cells. Each of those cells are individually alive on their own. You actually can take the cell out of your body and we can grow it in a we call it petri dish, right? Which is what these are. And the red liquid here, they are actually nutrient solutions. So if you take the cells out of the body and you give it the right conditions, give them the nutrients, put them at 37-degree chamber, they will be able to live outside of the body for some time. Okay? They will eventually die. Okay, even if you give them the optimal condition, they will eventually die. Just like they would eventually die in your body as well, right? Okay. Only some cells live a very long time. Not quite stem cells per se, but what kind of cells do you not get back if you lose them, like in the body? Brain cells, good. What's another one? For example, skin cell is easy to get, right? Okay, like we shed off our skin completely every couple of weeks, and then we get new ones. Okay, but there are some cells that we just don't get back if you lose it. Okay, brain cell is one of them, and there's another one. What's another important organ? What's that? Okay, liver maybe. Yeah. Heart, I heard the heart. Did you say heart? Yeah? No? Okay, maybe I'm just imagining the answer. Yeah, the heart cells. People who get heart attack, they don't, it's very hard to recover because you can't replace the damaged heart cells. Okay, so they tend to live for quite some time. All right. So that's that. Now there was this lady, her name was Henrietta Lacks, and she had cervical cancer in the 1950s, okay? And the people who were taking care of her at the time took a biopsy of her tumor. Okay, biopsy is like a sample of it, right? And then they didn't know at the time, and they just want to look at what happens if you grow them outside of the body. Okay? So unlike regular healthy cells, which I told you, if you grow it outside of the body, even if you give them the best condition, they will die eventually, right? But not for these cancer cells. If you keep on supplying them with the right amount of nutrients and the correct growing condition, they can divide and divide virtually forever. Okay, so that's when they found out cancer cells are actually immortal. They don't die. Okay, they can grow. They fill up the whole dish, and all you have to do is, take them out of the dish and then put them into different batches, and then now they will grow more and more and more. Okay, and so they found out this property of cancer cells actually allows people to study many aspects related to cells. In fact, they created this thing called a cell line. Okay? A cell line is basically immortalized cancer cells that can be used for research purposes. So the cell line was named after the person. Her name was, again, Henrietta Lacks. So they called that HeLa cells. Okay? And HeLa cells has been used in the study of things like tuberculosis, Ebola, HIV, as well as cancer treatment. Okay, so whenever you want to start creating a new therapy for whatever disease, you start at the cell level, testing them on cells. Okay, then you can move up to the model organisms, and then eventually you can move up to humans, right? If the data are good. So you can buy HeLa cells. Anybody can buy it. They're not cheap though. Okay, 20 microliter. Okay, 20 microliter will cost you $486 plus tax and shipping. Okay, so that's pretty expensive. How much is 20 microliter? Okay? It's so small that you can barely see it with your eyes, right? Okay, like this whole mug is 750 milliliter. Okay, so this is microliter, which is 1,000,000 times smaller than a liter, right? Okay, now it's not surprising that they are so expensive because, like, I've done this before. We order some in the lab and then you can just grow them forever. Okay, you grow them up and then you can freeze them at -80. And when you need more, you just store them and then you grow more. Okay, after you grow more, you can put some back into the freezer, right? So that's why it's like so expensive, because people usually buy it once and they don't have to buy it again, right? Okay. So companies who sell these things. Sorry, you had a question?
Is she believe that her cells were like given around the world without her knowing?
Yeah, so that's what I was gonna tell you next, right?
Oh.
So of course, companies who sell these cell lines made a lot of money. And actually, the woman died of her cancer, but her estate and her family actually sued the company, Thermo Fisher, over the HeLa cell line because they're saying they didn't get her consent in commercializing her cells, okay, and basically benefiting from her cancer, right? Making a lot of money from it. The company tried to counter-sue them. And eventually there was a little bit back and forth, but I didn't update the slides here, but I think ultimately her family won, okay? And the company had to pay them some money for, for the wrongdoing. So you can read more about it if you're interested. All right, next part of the cell theory. All cells, all living cells come from the division of pre-existing cell, okay? So here, nobody really knows where the first cell came from, okay? There are different theories, different beliefs, but we all know that in order to get new cells, they would have to come from the division of pre-existing cell. So here's a cell, and the cell can divide and become 2 cells. Each of those cells can further divide, and you can make more and more and more. Okay, so this process has a name. It is called mitosis. And we'll be learning a lot more about mitosis in the coming weeks. Okay, another question. See if you are more awake now. What do you think we're looking at here now? It's not humans, I will tell you, but like anybody can guess what this might be? Have you seen this before in high school, maybe? What's that? Tissue. Okay, what kind of tissue are we looking at? Maybe plants. Okay, get closer. Onions. That's right. These are totally onions. Okay. If you look at onion under the microscope and you stain them, okay, this is not the natural color. We stain them to highlight different features. That's what it is. Okay. Next time you eat onions, that's what you're eating. And this is an onion cell. And this is an onion cell that's dividing. Okay? It's splitting into 2. And these things are actually chromosomes. Okay, they're chromosomes. Which brings us to the last part of the cell theory. Cells contain hereditary material, usually in the form of DNA, which are passed on during division. Last time I asked you guys, how many chromosomes do we have? Do you remember? 46, that's right. So every time your cells have to divide, it would need to copy all of its genetic material so that each of the new cell will also have 46. Okay, you can't have 46 and then split into 23 and 23 and then 11.5, right? Eventually, you're gonna run out of genetic material, right? Okay. So 4 parts of the cell theory. Are we okay? All right. So cells are really small. You can't really see them. Like you can look at your hand, but you can't see the cell that's making up your skin, right? And in order to study cells, we need to use a microscope. Okay, so the first microscope was created by this person named Antony van Leeuwenhoek. Okay, he actually was a tailor, I believe, and on the side, he was just interested in making lenses. And one day he was looking through the lens, checking out some water, and he saw things moving in the water. So he called them animalcules. Okay, they are molecules that looks like animals, right? And then he got really fascinated. He kept on creating more and more lens, and he was looking at like everything that he could find in the house. And he did a detailed record, catalog, drawing everything that he saw. And he was telling people about it. But no one believed him, okay? Because no one was able to create the same lens. It was like, oh, this guy is like delusional. He's just making stuff up, right? So he got really angry, and then he invited prominent figures to go and see through his lens. So there were lawyers, there were politicians who got invited, and they were able to see the same thing, and they vouched for him, okay? But he never shared how he made the microscope. And this is what it looked like, ultimately when he refined his design. Nowadays, microscopes are very easy to get your hands on. You probably can go on Amazon and buy one for a couple $100. The ones we will be using in our lab looks like this. It's a light microscope. They can magnify things about 100 times, okay? So we're gonna be seeing a lot of exciting things. I'll show you bones. I have lungs, I have ovaries, I have testicles. Yeah, everything we will be looking at in the lab, okay? Not human ones, okay? They are like bunnies, okay? Rabbits. Okay, there are like higher power microscope that exist. These are able to magnify up to 2.5 million times, okay? So a lot more resolving power, and you're able to see much closer into and detail of the structure you're studying. And they are very expensive, okay? It could easily cost anywhere between $1.5 to $2.5 million for the whole package, okay? Nothing we can afford at the college. Usually the universities have them, or research facilities, hospitals, they would have them, okay? There are 2 kinds in general. You don't have to memorize these things. There's a TEM, transmission electron microscope, and scanning electron microscope. They do different things. This one gives you like a cross-section view of the specimen, while this one gives you like a three-dimensional views of the structure you're looking at. So depending on what you are interested in studying, the application might be a little bit different. So again, like I love to ask people to guess things. This is in the body. You have it, I have it. Where is it? It's wild guess, okay? There's no wrong answer. What do they look like to you, though? They look like hair? Yeah, they kind of do. You guys are no fun, okay? So quiet. These are in the airway, okay? Airway. Okay? When you breathe in a lot of pollutants and whatnot. So these things are in the airway. They will keep moving and then they sweep it back up so you can, cough it out or like blow your nose, right? Okay. People who smoke a lot, they develop what's called smoker's cough because the cigarette destroyed these little hairs in the airway. They can't get the stuff out naturally, right? So they have these heavy, like really phlegmy cough, right? To physically try to cough out the stuff that are trapped. Yeah, smoker's cough. Okay, how about some concept check question? Which of the following is a concept of the cell theory? I'm gonna take a seat while we do that. That's good. Yeah. Don't be subtle about it, guys. Like I can doing it so like secretly, like just put out the fingers and then which way found it. What is it at the back? Yeah, that's good. That's good. Yes. Yes. Good. Most of you got it right, right? Or a cell is a basic structural and functional unit of living things. Okay, the other three are not correct. Organelles are not basic unit of life. Cells are. Simple cells arise spontaneously from rotting vegetation. No. To get new cells, we need to have division of pre-existing cells and not just complex organisms. All living organisms are composed of cells. How about here? This one should be pretty straightforward. Good, good. Yes. Good, good. Very good. Okay. They have the cytoplasm, they have genetic material and the membrane, but nucleus, which we haven't talked about, is not necessarily present in all cells. All right, cell membrane and the nucleus. If we take cells and we separate them into two categories, the categories are prokaryotic cells and eukaryotic cells. Let me just put it on the board here. So eukaryotes, or you can say eukaryotic cells, they mean the same thing. Typically, eukaryotes are much bigger, they are more complex, and they are more sophisticated than prokaryotes. You can remember P is for primitive. Okay, so the prokaryotes are more primitive. Okay, they are smaller, they are simpler. Okay. The defining feature that distinguishes the two is the presence of a nucleus. Eukaryotic cells have a nucleus. Okay, so there's like a circular structure that is inside the cell. It's like a spherical structure inside the cell. which holds genetic material. So our DNA is packaged inside the nucleus. Okay. So eukaryotic cells have a nucleus. Prokaryotic cells do not. Okay. In addition, eukaryotic cells have many different types of organelles. Okay, so all these things that you see listed here, all of them are organelles, and each will have a different function. We'll be learning all about it. Okay, so organelles are specialized structure inside a cell with specific function. So animal cells are eukaryotic cells. Plant cells are eukaryotic cells. And again, I just want to emphasize animal and plants are not the only two types of cells that exist. There are fungus, there are protists. Those are also eukaryotic cells. And you can't read my writing. It says specialized structure with specific functions. On the other hand, prokaryotic cells, they are much smaller. They are much simpler. Okay? They do not have nucleus, and they have only a few types of organelles. Bacteria are prokaryotes. Do you guys know the name of any bacteria? Anything that you've heard of from the news? Anytime there's a recall for like lettuce or something like that, what was it? The almond milk that they recalled recently, right? What are some bacteria that you know? Yeah, there's Salmonella. Anyone else? E something, you must have heard of it, right? E. coli, yeah. Okay, so all these are bacteria, right? Okay, and they are all prokaryotes. Remember, bacteria are much smaller, right? Earlier, I was telling you, animal cells are measured between 10 to 100 micrometers, but bacteria, they are 1 micrometer, right? So much smaller. Okay, so here is the cell membrane, and in case you forgot, the cell membrane is primarily made up of these things. These are called phospholipids. Okay, remember we learned this on Monday. The phospholipid has a hydrophilic head and hydrophobic tails, right? I told you it's like detergent. The head binds to water and the tail stays away from water. And the cell membrane is actually made up of 2 layers of these things. I told you it's like a peanut butter sandwich. Right? The bread are like the hydrophilic heads, and the peanut butter in the middle would be the hydrophobic tail. Now, the cell membrane is selectively permeable. If something is completely permeable, then it means everything can pass through. Right? If something is non-permeable, that means nothing can pass through. Selectively permeable means it allows something to go through while blocking others, okay? Which is what you want, right? You don't want a cell membrane to be completely permeable, because then harmful things can come in. Okay? You want it to be selectively permeable. Keep the harmful stuff outside and then keep the nutrients and the resources in the cell. Okay, so it's not like freely warming back and forth. If we take a closer look at the cell membrane using one of those high power microscope, you will see that this is the cell membrane. The two black lines are actually the heads, the hydrophilic heads. And then the white gap in between, that's where you have the hydrophobic tails. Okay. So it's not a single layer, it's a double layer. I should point out that the cell membrane is not just on the surface of the cell. Several organelles that we will be learning are also made up of the same material. So you have some membrane inside the cell as well. Okay. All right, that's enough of me talking. Let's take some class notes, okay? Now I'll show you what we are drawing, so you plan accordingly. This is what we're doing, okay? This is what we will be doing, all right? You only have to do it once. I gotta do it 3 times, okay? In the afternoon, I gotta do it again because I have another class, but I love it, okay? So, if you have a piece of paper, I suggest you start here, and then we're gonna work our way down. All right? People are telling me they run out of space. Right? So start it here, slowly go down, and then we will add some stuff on the side as well. Okay. It would be ideal if you have some color to draw some highlighters or something. If not, that's okay. Just go black and white for now. And maybe when you go home, when you're studying, you can watch the video and draw it again nicely. Now, I'm not going to ask you to draw anything on tests or anything like that, okay? But, you know, over the years, people tell me it is easier for them to understand and to learn it if you're drawing it together with me, okay? Rather than clicking through the slides, it's just like information overload, right? All right, so here we go. So let's start with a nice round circle. Okay. Nice round circle. By the way, we are drawing a eukaryotic cell, okay? So what is one thing that eukaryotic cells have that prokaryotes don't? The nucleus, that's right. And this is the nucleus. All right. Now I gotta say, right? There are exceptions, right? I have been trying to tell you this since day one. Not everything is necessarily gonna fit perfectly into the categories that we create. All of our cells are eukaryotic cells, but your red cells, for example, they don't have a nucleus. Okay, and we can learn why next semester if we meet again. Okay, so here this is the nucleus, and I'm just gonna put it right here, nucleus. So the nucleus is wrapped by a membrane. Okay, and I'm not gonna color the whole thing because I want to look inside the nucleus in a bit. So I'm just gonna wrap like maybe 70% of this guy. Okay. You guys know the chocolate like Ferrero Rocher with the golden wrapper with hazelnut? It's like that, okay? The wrapper is the membrane, but I'm opening up a little bit of it so I can see the inside, okay? So this here is called the nuclear envelope. Okay? This here is the nuclear envelope. And it surrounds the entire nucleus. Of course, I'll post a note as well. So there's no need to take pictures. On the nuclear envelope, there are some openings. Little openings here and there. And these openings, they are called nuclear pores. So the nuclear pores allow material to enter or leave the nucleus. So if something needs to go in, they got to go through the pores. Same thing, if something wants to come out, you got to go through the pores. So far, so good, I hope. Now, inside the nucleus, there is an other spherical structure. Okay, just like the Ferrero Rocher, if you're following the analogy, there is a nut inside, right? Okay. It's pretty much like that. So there's an other circular structure here, and that is called the nucleolus. By the way, like I sometimes people get a little bit frustrated. It's like, why are you teaching all these things to us, right? Like, it's not me. This is a ministry standardized curriculum. If you go to pre-health program in other colleges, you'll be learning the same thing as well, okay? So I'm just the messenger. Now, the nucleolus is an organelle, and its job is to produce another organelle called ribosome. And we'll get to that later. So you make the ribosome will leave the nucleus through the pores, through the openings. So what's the purpose of a nucleus? Well, if you wrote down the notes earlier, you might remember the nucleus is where we will keep our genetic material, right? Yeah, our DNA. So let's go over here for a second and talk about what exactly is in the nucleus in terms of genetic material. Last class, we learned about DNA. Okay? And DNA looks like this. DNA looks like this. It's a double helix, and then these lines I'm drawing, those are the ACTG pairing with each other, right? A pairs with what? T, right? Very good. C pairs with G, right? So this is DNA, but this is naked DNA. That's what it's actually called. Okay? It's naked because it's not associated with anything. It's just DNA by itself. And we generally don't find naked DNA on its own. Okay? It's very exposed. It's very vulnerable. It can get damaged easily, right? And it's really, really long. So we can't really fit all of the DNA inside the nucleus unless we package it nicely, okay? Each cell has 2 meters of DNA. If you lay it all out, the total length is 2 meters. And we need to pack 2 meters of it inside this tiny structure called the nucleus, okay? It would be same as, like number-wise, it would be same as packing 4 kilometers of sewing thread into a tennis ball. Okay, so in order to do that, you really need to wind it up, right? And so what your body does is it takes these DNA, and then there are these packaging proteins. These are packaging proteins. Packaging proteins. And you basically take the DNA and then you wound them around these packaging proteins. Okay, wound them up, and so on and so forth. The best way to visualize this is to think of a yo-yo. Okay? The rope is like the DNA, and then the yo-yo, it's like the packaging protein. Right? In order to fit the rope nicely, you wound the rope around the yo-yo, right? Okay? Except over here, you have multiple yo-yos and you're wounding a very long rope all over the place. Okay? So this stuff right here has a name. And when you have the proteins and the DNA all packed together, we call this chromatin. Chromatin. And that's what is found inside the nucleus. Okay, these are chromatin. Now, some people ask, well, how about chromosome? Right? What's the difference between chromatin and chromosome? Well, it turns out it's the same stuff, but the chromatin can actually pack even closely together with more packaging protein. So it loops back more and more and more like this, and it forms a really, really, really condensed fiber. Okay? And sometimes you can actually copy them, and then you have your chromosome. Okay, if you search up chromosome, or you've probably seen it in a movie, they usually come with like shapes of X. Okay, and that's what it is. It's just super compact chromatin. So normally, listen for a second. Normally, when a cell is not dividing, it's the genetic material is going to be in the form of chromatin. But when you need to move the genetic material around, such as when you're dividing, then you need to pack it up even further into chromosome, so it's easier to move around. And then when you're done moving them around, you can go back to chromatin. So this is just like compacting even more. It's like you moving, right? If you move, you got to pack your stuff in a suitcase, and then you can move it easily around. It's the same content. Okay? And once you're done moving, you can take it back or out. You can unwound. So that's the difference between chromatin and chromosome. These two things? Yeah, that's a good question. Yeah. They are connected. Okay. So like this is one chromosome, and then this is another one that's connected to it. So generally they form like an X. Okay? But we'll talk more about this later on. For now, I just need you to understand, the difference between DNA, chromatin, and chromosome. Are we together? Just give me a tiny nod. Yeah. Any questions? Have fun here. Okay. So can everybody visualize the nucleus and everything associated with it? Yeah. Okay, maybe just one more analogy for those of you who are having trouble. You don't have to copy this down, okay? You can just watch, because I'll erase it afterwards anyways. It's not important. Okay? If you think of like an avocado, okay? Think of an avocado with the seed inside. Okay, and then we have the meat here. There we go. Okay, and then we have the skin. Right. If the avocado is like the whole nucleus, then the seed would be the nucleolus. Very good. Okay. And what would you find in the meat? Chromatin, right? Okay. And then the skin, the shell, whatever you want to call it, that would be the nuclear envelope. Is that okay? If you take a toothpick or fork and then you poke holes on the avocado, the holes would be like the nuclear pores. Okay? All right, let's draw more. Now, this next part is a little bit confusing. Stay with me. Hopefully it will all make sense in the end. The nuclear envelope actually does not stop around the nucleus. Okay? The nuclear envelope will actually continue to extend outside of the nucleus, okay, and form these networks. Okay, you can just like randomly draw some ribbons like that. And they, can even extend further out like that. Okay. So I'll write for you here. Forming a network of membrane. Okay, so the ribosome, let's take a closer look at them. The nucleolus makes the ribosome, and the ribosome will leave the pores and go into the cytoplasm. Right? Some of the ribosome will be on its own in the cytoplasm. Okay? So these are free-floating ribosome. Some will attach onto this network of membrane. Okay. They could be on their own, floating around, or they could be stuck on this network. So more on the ribosome first. It says down here it says ribosome attached to membrane network. Now I want to zoom in on one of these ribosomes, okay? I want to zoom in. They are really small compared to other organelles, but if you zoom in and you take a closer look at them, they are actually like a hamburger. Okay? So there's the top bun, which is bigger, and then there's the bottom bun. The top bun is called the large subunit. And as you can probably guess, The smaller one is called a small subunit. The ribosome is made up of protein and RNA. And the function of the ribosome is to produce proteins. Another question for you. What are the monomers of protein? We wrote down last time. What are the monomers of protein? Yeah, what is it? You got it right. Huh? Oh yeah, okay. It's made up of CHO, but what are the building blocks of the protein? Yeah, amino acids, right? Okay. So the ribosome is responsible for connecting the amino acids together. Okay. The ribosome, in fact, will use the instruction from the DNA to build the protein. Okay, so that's what the ribosomes do. They produce proteins. Okay? They produce proteins. Why don't we take a moment to pause here, have a break, and we'll do more after. Okay, it's a lot of things for you, I guess. We will resume at 9:35. Okay, 7-minute break today instead of 5. Okay, enjoy it. All right. So we were talking about the ribosomes. And like I said, the ribosomes are made in the nucleolus. They can come out to the cytoplasm where they're floating by themselves or they are stuck to the membrane network. Okay, so we already talked about the free floating one. They make proteins by connecting amino acids together. And now let's shift our focus to these networks. There are two kinds of network. I'm just going to label it for you, and then we'll talk more about it with the PowerPoint slides. We're almost done with this picture, okay? So over here, the network that contains the ribosomes, if you look at them under the microscope, people think that they look a little bit rough, okay? So the name for these ones is called the rough endoplasmic. Endoplasmic means like within the cell, reticulum. Okay. Short form is RER. Rough endoplasmic reticulum. Yeah, so these networks with the dots, with the ribosomes, the whole thing is the rough endoplasmic reticulum. Okay. You okay? Question? No? Yeah?
All the purple line and good.
That's right, the purple line with the dots. Together is the rough endoplasmic reticulum. And over here, it's an extension of the network, but they do not have the ribosomes on them. So when you look at them under the microscope, they look a little bit smoother, right? So people call it the smooth endoplasmic reticulum. Okay. With the ribosomes and without. let's take a look at what the rough ER does. I'll put it here. The rough ER. The details are in the PowerPoint slides, but like, I recommend you memorizing like the function of the organelles with just one line, okay, to start anyways. All right, like to forget about all the details, just remember the basic function of it, and then you can memorize more things. For example, if I say ribosomes, you will tell me they produce proteins. Exactly. Right? If I say nucleolus, then you will tell me they produce ribosomes. Okay, so just try to memorize one thing first before moving on. Now, in case you forgot, your second quiz is going to be done online, right? So you will have some time to remember the things, and you know, it's okay to access your notes as well for quiz number 2. Okay, I'll tell you more about it later. All right. Rough ER, what does it do? It works with the ribosomes to produce and package proteins. Now, let me try my best here to show you how it looks like. It's really hard to show you with drawings, but I will try. Okay, maybe watch first and then draw it out later. So I'm zooming in on one of these channels. Okay, and here is the hamburger. They actually do look like this, okay? If you look at it under the microscope, they do look like that. There is the ribosome. And the ribosomes, they make proteins, right? So it will connect these amino acids together. These little circles are amino acids. And then it will put the protein inside the rough ER. Okay, so that's the first step. Okay. Ribosomes synthesize proteins into the rough ER. Now, of course, we know proteins are gonna fold up, right? Like into my green little ball and the pink ball thing, right? So it's gonna fold up into a protein like this. So that right here is the protein. And this next part is hard for me to show. So maybe just stop copying and watch first, okay? Now this protein is gonna move towards the edge. Gonna move towards the edge. And then what's gonna happen is, I'll just draw the tip here for you, and hopefully it will make sense. Okay? So once it moves to the edge, once it moves to the edge, it's gonna start to pinch off. Okay, this is the protein. And then it will continue to push towards the edge until eventually it completely detached. Can you visualize that? Maybe? Right. So the protein will move towards the edge, and then it will cause this part to like kind of pull out, and then eventually it just pinches off. It's like blowing a soap bubble. Right? When you blow the soap bubble, it comes out, and then it bubbles off, right? That's exactly what happens for real. Are we okay? I'll post an animation for you to watch, okay? I don't like playing videos in class. It's waste of time, okay? You can watch, you know, the 6-7 minute video at home at your own leisure time, okay? So what happens in the end is, not only are we making the proteins, but we are able to package it. So this is a vesicle containing proteins, and more than one usually, will hinge off from the rough ER. The free floating ribosomes, they can also make proteins, but those are not packaged. Okay. Once you make them, they're just, there. You're not going to put them around a bubble. Okay. So that's it for the time being. Right? We'll come back to this drawing in a bit, but I want to go through a few slides with you first. Now, while I have your attention, I just want to show you one more time in case you forgot about this. If you go to the homepage on eCentennial, the news has been updated, right? So that's the concept track question. But if you scroll down, okay, to the Wednesday news from last week, okay, this is the information about the quiz. Right? We will have 15 multiple choice questions plus 5 short answer questions. Some of the questions will be same as the study guide. Okay, I literally copy and paste and put on it. Okay, so please make sure you do the study guide. Do you guys know where to find the study guide answers? Yes? Good. If you don't know, see me after class before you go home. There will be 40 minutes for the quiz, so please arrive on time, okay? There is going to be a seating plan for quizzes. We always do this, okay? So when you come in, just take your pencil and eraser out. You will leave the bag on the side of the classroom, the lab, okay? Leave it on the bench where you do the labs. And then there will be a seating plan on the screen, and then you just find your seat and sit there, okay? It will be 40 minutes, okay? Don't be late. If you're late, you're not going to get extra time. Again, you need a pencil and an eraser to do the Scantron. And most importantly is this video. Okay, watch it. Okay, if you can't find it later on, just email me. I'll send you the link. Okay, but that's very, very important. Okay, it's not difficult. Okay, I swear it's not difficult. The quiz. All right, so a lot of these slides have the information that we just draw. So I'm not gonna go through them again. You should read them on your own when you have time. Okay, so we talk about the nucleus, we talk about the envelope and the pores, and this is the professional drawing. Okay, you have the nucleus here. You see the pores. Okay, what are all these yellow dots? Take a wild guess. The ribosomes, that's right, okay? So sometimes they are stuck to the network, making it rough, ER. And then you have more network that do not have the ribosomes, and we call them smooth. Okay. This is the DNA I was telling you about. They wrap around the yo-yos, the proteins, and then they become chromatin. But they can further condense, they can pack even more and become the chromosomes. Huh? We talk about the nucleolus, which by now you should remember they make ribosomes. And the ribosomes do actually have 2 subunits, like a burger. They are usually separate, but when they make protein, they will come together. This is what they look like under the microscope. It's really small, but if you zoom in more, you will see that it's actually 2 things. So these are ribosomes that are on its own. These lines, they are the ER, okay, with the ribosome stuck on. For tests and quizzes, I will only show you cartoon diagrams to identify. Okay, I'm not going to show you one of these pictures, actually what you're looking at. Okay, that's not fair. I'll show you the burger though, and you should be able to tell me it's a ribosome. Okay, only cartoon pictures. And there are lots of these things in the study guide for you to get familiar with. Okay, so I'm just gonna read this to you. it's the same thing I've been telling you. Ribosomes are made in the nucleolus and exit into the cytoplasm. In order to exit the nucleus, you need to go through the pores, the holes. Some ribosomes exist freely in the cytoplasm. They're just floating around, okay, like jellyfish in the ocean, while others are clustered and attached onto network of membranes. We all know those are the rough eRNA, right? Rough endoplasmic reticulum. Okay, so I want to spend a bit of moment and talk about this concept. This is a very important concept that we will continue to explore for the rest of the course. It's called the central dogma of biology. Dogma is just like a fancy word for theme, okay? Central theme of biology. Okay. So DNA contains the instruction to make proteins. That much we know, right? From Monday's lesson. But DNA is very, very long. I told you we have 2 meters of DNA all crunched up together in the nucleus. It's not possible to take the DNA out of the nucleus. Like it won't even fit through the pores. Okay? And even if we could take it out of the nucleus, we actually don't want to do that. We don't want the DNA to be damaged. Okay? If you damage the instruction to make the protein, as we learn, you can make the wrong protein, wrong shape, wrong function, right? So we want to keep the DNA in the nucleus. Whenever we want to make protein, you actually will make a copy of the DNA in the form of RNA. Never mind what the M stands for now, okay? Just know that we're going to make a copy of the DNA in the form of RNA. When you do that, when you copy it, this is called transcription. Transcription. We're not going to copy the whole thing. We are just going to copy the section that we need to make a specific protein. Okay. Think about it like this. At the library, there is this big book of test questions. Right? They don't let you sign it out. Right? And they don't let you use your phone. So what are you going to do? You photocopy it, right? Okay. Do you photocopy the whole book? No. You only photocopy the section that you need to study for your test, right? That's what happens here. You're not going to copy all 2 meters of DNA, only the section that is needed to create a protein. Okay. Some of you might have heard of the term genes, right? Okay. That's what this is. You're copying only one gene for one protein. Once you make the RNA, right, the copy of the DNA, the RNA will exit through the pores and it will go into the cytoplasm. In the cytoplasm, the ribosome, the burger, is going to grab onto the RNA and it's going to read through it. Okay, a lot of textbooks describe this as the burger and spaghetti model. Okay, think of the burger like moving through a noodle. Okay, and along the way, it's going to read the instruction and make the protein. This process of creating the protein from the RNA is called translation. Okay. So that's how information flows in all known life form. Okay. There are some situations where it goes the other way, but those are exceptions. Okay. For the most part, information goes from DNA to RNA to protein. Okay. Now let me remind you, DNA and RNA, they are both nucleotides. So when you are making a copy, it's called transcription. When you transcribe something, you're just copying it using the same language. RNA is made up of nucleotide, and proteins are made up of amino acid. So when you go from RNA to protein, it's a different language. That's why it's called translation. Okay. So transcription happens in the nucleus and translation happens in the cytoplasm. Okay. All right, very quickly. Which of the following is a prokaryote? Yep. Good. Try again. What was that? Try again. Eukaryotes. Yeah. Good, Christine. Not quite. Henson, good. Okay. So prokaryotes are bacteria, right? Okay. And which one is the bacteria here? E. coli. Liver cell, liver is in us. That's a eukaryotic cell. HeLa cell, that's a cancer cell from the cervix, right? Okay. That's prokaryote. Amoeba, also prokaryote. Okay, how about this? Good. Anyone else? Yeah. Yeah, if you are with me the whole time, you should know. Nucleolus, they produce ribosome. Okay. A is the answer. Okay, so we have time to talk about the endomembrane system. In fact, we already started talking about it when we drew the picture. So what is the endomembrane system? Endo means within. Endo. So this is a system made up of membranes, which are found within the cell. I told you earlier today, the cell membrane is not the only place where you'll find a membrane. You will have more membranes inside the cell. Together, the entire system, the entire endomembrane system is responsible for the synthesis, the production, processing, digesting of lipids and proteins and other large molecules. Basically, they form like a supply chain almost, okay, of breaking things down, building things, packaging them, and sometimes even recycling and destroying unwanted things. Okay? So it will make more sense once we go through the whole process. When you go back to this slide, it's going to make more sense. Here are all the components of the endomembrane system. We already learned about the nuclear envelope. We know the nuclear envelope extends outward to form the rough and smooth ER. We know the rough ER makes protein and packages it. We haven't talked about the function of these things yet. Okay, we will in a second. So here it is again. We know this already, so I'm not going to spend too much time on it. The rough ER is rough because it has clusters of ribosomes on them. And this is what I was trying to draw for you. Okay, let's take a look again. We have the ribosome on the surface, and it's making the protein directly into the ER. The protein folds up and moves towards the edge where it will butt off as a vesicle. Okay. Now, this vesicle is not finished yet. The contents inside the vesicles, those proteins that we just created, they need to be modified a little bit. So there is another organelle that does that. But before that, let's take a look at the smooth ER. Okay. In a picture, how do you tell the difference? Well, you can look for the dots, like dots or no dots. And also the smooth ER tends to be further out from the nucleus. Okay. So they have no ribosome attached onto them, and their function is to make lipids. Okay. Rough ER makes proteins, smooth ER makes lipids. And we learn about different kinds of lipids, right? There are phospholipids, there are steroids. So whenever you want to make these things, you have to do it at the smooth ER. For example, we learned that steroids such as testosterone and progesterone and estrogen, they are made in the ovaries and testicles. So if you look at the cells of the ovaries and of the testes, you will find more smooth ER in them. Okay. I need you to understand that not like all the cells in the body not all of them are gonna have the same number of each type of organelles. They won't all have the same amount of like rough ER or same amount of smooth ER. It depends on what their functions are. Okay? Some cells will have more of certain types of organelles than others, right? That would be more appropriate for their function. So these cells would have more smooth ER because they need to make more of the steroids. Another function of the smooth ER is that they help break down fat and detoxify drugs. Right? It turns out when you take drugs, like not necessarily like illegal stuff, right? Like regular medication, if you take antibiotics or something like that, okay? Whenever you take medicine, whenever you have drug in your system, your liver cells will actually make more smooth ER to help cope with the medication, to help metabolize the drugs, okay, and break it down. Right? So a couple of things to know here. Not only are the number of organelles different for different cell type, for the same cell type, they can actually change the amount of organelles depending on what's needed. Right? If you need to metabolize drugs, no problem. We make more smooth ER. Okay? But once the drug is out of the system, then maybe we don't need as much anymore. Okay. So on the quiz, like I can describe the function of a particular cell type, and then you can tell me what kind of organelles do you expect to find more of. All right. Okay, so let's go back to our picture and finish it up. Let me zoom out a little bit. We have not drawn the cell membrane yet, so I'm going to do that. Okay, I'm just going to put a boundary here. This is the cell membrane, okay? Doesn't really matter. This is like an arbitrary. Okay, so this here is the cell membrane. Right? So like this whole thing is the cell, and then that's your nucleus, and then a bunch of other stuff, right? By the way, hands up if you have your study guide with you for lecture 3. 1, 2, 3. Okay, can you guys bring it for next class, please? All right. The study guide for lecture 3, Monday. Okay. We'll label something from there. Okay. Let's talk about this vesicle. Okay, this bubble that is freshly made from the rough ER. We have a protein in it, but it's not quite ready yet. So this thing is actually going to go to another organelle. And that organelle is the Golgi body. Right? It's several membranes that are stacked on top of each other, kind of like pancakes. This is Golgi body. And sometimes textbooks, they might call it Golgi apparatus. It doesn't matter. For me, you can use either one. Okay. So the tagline for you to remember for Golgi body and Golgi apparatus, in terms of what it does, is that it sorts, modifies, and repackages, I guess, proteins. You can think of it as like the sorting center of a post office. You have all these packages going into the post office. They got to sort it so they know what goes where. Sometimes if the package is a little bit broken, they got to repackage it. Okay. So the vesicle will come, and then it will enter the first membrane of the Golgi body, and it will actually get passed forward, and sometimes it will go back. It just go back and forth a couple of times, and eventually we will get a new vesicle, and then the protein inside is going to be modified. And I'll use a different color to show that it has been modified. So this is a modified protein. Now you don't have to really know the modification that's happening, but in case you're curious, sometimes they have to cut the protein if it's too long. Sometimes they have to add certain tags to the protein so it knows where to go. And sometimes you even have to add like sugar onto the protein to make it into a sugary protein. Right? So there are a lot of things that could be modified. The point is the vesicle from the ER is going to come here and it's going to get sorted, it's going to get modified and repackaged. Now we're almost there. But the next thing we want to know is what's going to happen to this protein. Okay, let me just make the cell membrane a little bit bigger so I have more space. Okay, what's going to happen to this protein? Well, there are a couple of places it can go. Number one, this can actually go to the, give me a second here. This can actually go to the surface of the cell and you can release it. Okay, you can release the protein. Okay, number one, protein released out of the cell. Okay, so maybe you need the protein right away. The vesicle goes there, shoot it out of the cell, and maybe go into your bloodstream or something. So that's one possible path. Second possibility. Now, I told you the cell membrane is primarily made up of phospholipids. There are a little bit of cholesterol, but actually, sometimes you might have proteins in it as well. Okay, so proteins become part of the cell membrane. Okay, sometimes you don't need it right away. You don't need the protein right away. Like I'm just making it just in case I need it for later. Okay, so number three, stayed in cytoplasm until it is needed. Right, so you don't have to make it on demand. You already made it. You have a stock pit of it. And when you need it, you either release them or you use it directly in the cytoplasm. And the fourth possibility here is that it can becomes another organelle. Okay, I think this is the last one we'll do today. Becomes a lysosome. Becomes a lysosome. Okay, so this guy also looks a little bit different, and then it has like stuff in it. We'll go through the lysosome in the slides. So to recap, I'll let you finish copying first, and then I'll recap it for you. Okay. So to recap, if we want to make the protein, we have to start all the way in the nucleus, right? There's the DNA that tells you how to make the protein. But we can't take the DNA out, so we make a copy of the DNA in the form of RNA. That's right. We make a copy of the DNA in the form of RNA. This is called transcription. Okay. So the RNA that we make will go out to the cytoplasm, and it can either be used by the free-floating ribosome or by the ones that are attached to the rock ER. The rock ER, if we follow that, it will use the instruction to make the protein. The protein will butt off in a vesicle, but it's not complete. It needs to undergo further changes, further modification. So this thing will go to the Golgi, and after it gets modified, it can go to one of these paths. Okay, that's basically what the endomembrane system is. It's a network that does all these things. Okay, of course, don't forget the smooth ER is also part of the endomembrane system. Okay, but instead of making protein, it's important for making lipids. That's right. Okay, lipids. All right. Okay, so let's talk about the lysosome. The lysosome is the recycling depot, if you would, okay? It's one of the possible paths of the repackaged protein. But in this case, the lysosome contains a lot of digestive enzymes. I don't know if you remember, but enzymes are proteins for speeding up reactions. Okay. Every reaction that happens in the body will need an enzyme. Okay. So there are a mixture of these digestive enzymes that are inside the lysosome. So when your cell ingests some kind of food particle, for example, it will merge with the lysosome to break it down. If there is a damaged organelle, it's too old or is not functioning properly, the lysosome will come and it will break it down. Okay? So it breaks down worn out molecules, it breaks down macromolecules, and you can unlock the nutrients and recycle them. It's also possible for a cell to purposely release the enzymes from the lysosome and destroy itself. This is called apoptosis. Programmed cell death. It purposely destroys itself. Now, why would a cell do that? Well, sometimes it's part of a normal developmental process. When we are developing as a fetus, we actually have webs around between our fingers. Okay? So you can see this is 51-day-old fetus. There is the web between the fingers. There's the web, right? So of course we are born without webs. Our fingers can move freely. So what happens to all the webs, all the cells in the webs? Well, they all undergo apoptosis. Okay, the cells deliberately release the contents from the lysosome and destroy it. Okay, so sometimes it's a normal developmental process. Other times, as we'll see later on in the course, a cell is not functioning properly. A cell is dividing too fast. Okay, maybe it's precancerous. Then you want to destroy that cell. So it triggers apoptosis. It kills the cell off before it's totally out of control. Okay, so it releases all the contents from the lysosome. Now, I'm not going to test you on this, but very briefly, okay, so like I'm just going to put an X here, as in you don't have to memorize it. But the enzymes inside the lysosome, there are many different enzymes. And these enzymes, as I tell you, they are proteins, which means the instruction to make them is in the DNA. And we should all know by now, DNA can change, DNA can mutate, right? So a person can inherit mutated DNA, which cause them to be missing some of these enzymes. in the lysosome. Depending on which enzyme is missing, the disease that you get out of it is going to be different. But collectively, these group of diseases, they're called lysosomal storage disease. For example, in Pompe disease, the enzyme in the lysosome for breaking down glycogen is missing. You remember glycogen is storage of energy, right? In muscle and in liver. So if you don't have the enzyme to break down the glycogen, they start building up in the muscle, and it causes the muscle to become weak. This is, interfering with the muscle contraction. And you can imagine if the weakened muscle involves in breathing or heart contraction, then the person is going to have, cardiovascular problems and breathing problems. Tay-Sachs disease is one of the diseases you can screen prenatal, using genetic material from the child, and you can do it as early as 21 weeks. And this one is missing a lipid digesting enzyme in lysosome. So these lipids build up in the brain and they interfere with brain development. Okay, usually it's fatal. Okay, after the child is born. So, the lysosome is actually quite important. All right, just 2 questions here, 2 more concept check, and then we're gonna go. Do you remember the answer for this one? Okay, that's one of them. Good. What else? Good. One of them, yeah. Good. There are 2 answers. That's right. Okay, so ribosomes are made up of RNA and protein. Okay, RNA and protein. There are also 2 answers here. My caption is in the way. Okay, good try. Yeah, that's right. Yes, for one of them. Not that one. Yes. Anyone else? Okay, so to break down molecules, we need the lysosome, right? We literally just talked about that. But what about detoxifying toxins and waste? What organelle does that? Do you remember in the liver, you're gonna put drugs? Yeah, that's movie, right? Okay, small. Okay. So on the written quiz, all questions will only have 1 answer. Okay, 1 correct answer. For the online one that you will be taking for quiz 2, some questions will have multiple correct answers. Okay, that's it for today. Please study hard for the quiz. They do tend to get harder as we progress through the semester. So get all the grades you can while you can.